Hello, everyone. This is a demo of the single-cell RNA-seq analysis pipeline within the Bridge Horizon platform. I'm going to run through the pipeline, how to configure the samples, as well as some of the advanced settings, and then we will go through the outputs as well. So in order to launch this pipeline, you're going to click Launch Pipeline, which will take you to the sample configuration page, as well as configuring some of the advanced parameters within the pipeline. So first, we will discuss how to configure the samples. So to configure the samples, you're just going to select the folder that contains your FASTQs. So I'm going to be running the pipeline on the human single-cell RNA-seq FASTQs. I'm going to select that folder, which contains the two FASTQs, and drag and drop. Perfect. So as you can see, it automatically detected two different samples within this folder, and then gave it unique experiment IDs. Now, because I want these two FASTQs to be analyzed as part of the same experiment, I'm going to give them the same experiment ID. And then what the pipeline will do is integrate them so that when you do visualization or differential gene expression analyses, both these samples will be visualized and analyzed together. And this is appropriate because they were part of the same experiment. Alternatively, you could give them separate experiment IDs, at which point these two samples will be outputted in separate folders with the experiment ID, and then they will also be analyzed independent of each other. So they are both 10 by V through chemistry, so I'm going to select that over here, and they're both human, so I'm going to select human over here. Okay, once the sample shield is configured, I'm going to click Done over here. As you can see, now we have two samples configured. One of the main required settings in order to run the pipeline is the resolution parameter. And as the description details below over here, this parameter controls the granularity of clustering. So as you increase the resolution parameter, you're going to get more clusters, or your cell populations will be split into more clusters for all downstream analyses. The default is 0.5, but depending upon your cell populations, you may want to increase or decrease this resolution. And you can do that by just changing the numerical value as I just show over here. I'm going to keep 0.5 because that has worked across multiple data sets. So I'm going to set the default as 0.5. Or I'm going to keep the default as 0.5. Now, before we run through the pipeline, I'm just going to go through some of the advanced settings over here. So one of the main steps in any single-cell RNA-seq analyses is the annotation. So we have a automated annotation engine built in to this pipeline. You can select the appropriate references for this annotation engine over here. As you can see, we have multiple organs that have been pre-configured for automated annotation. This is PBMC dataset, so I'm going to keep the default as it is, which is PBMC. And then some of these parameters just control the length and width of the plots that will be generated using these automated annotations. Just to provide some context, these numbers represent dimensions in inches. And you can obviously change them as in these boxes over here. Now, the people that do single-cell RNA-seq analyses tend to be quite sensitive to the color palettes that are used for visualizations in UMAPs, violin plots, bar plots, and so forth. You can change the color palette over here by entering any of the colors that you find appropriate. This is the default color palette that I've configured, but obviously this can be adjusted to your specifications. This over here, this setting over here controls the resolutions that you want to test when you're doing clustering. So as you can see, I'll be testing resolution parameters 0.125 all the way to 1.2. You can obviously adjust this as necessary. And what the setting will do is that it will generate a UMAP that is built using these different resolutions so that after you run the pipeline, you find that the resolution of 0.5 is not appropriate for your data set. Using that view map, you can find the appropriate resolution and change that setting over here to then rerun the pipeline. So this is incredibly helpful when you're done running the pipeline. The next set of settings control the parameters that are used for differential gene expression analyses. So the first slider input controls the log to full change threshold that must be met by your genes for them to be considered for differential gene expression analyses. Point 2 is the default that is recommended by the people that develop Seurat, but obviously you can change that to your specifications. The minimum cell per group, so this parameter controls the minimum number of cells that must be present within a given cluster in order for it to be considered for differential gene expression analyses. So if you have less than 5 cells per cluster, those clusters will be dropped for DG analyses. This controls the PCT, or the minimum number of cells that must have expression of a specific gene for that gene to be considered in DG analyses. So what this simply means is that for a gene to be considered for DGE analyses, at least 20% of the cells within a specific cluster or a subpopulation of cells must be expressing that gene in order for it to be present within your DGE list. So if you want to look at high confidence genes that are expressed in the vast majority of population within your cell cluster, you can obviously increase this threshold as necessary. This over here controls the p-value threshold, or the FDR adjusted p-value threshold for your DGE list. And again, this last radial input over here controls which data you want to be using for your DG analyses. Your data slot over here contains the raw gene counts, which is recommended to be used for DGE, but obviously you can adjust that as necessary. The rest of the plots control the dimensions of the visualizations that are generated after you complete DG analysis. The rest of the inputs over here, or settings, control your clustering and integration steps. This step over here, or this setting, controls which algorithm you want to use for doublet detection. Obviously, when you do single-cell RNA-seq analyses, it's possible that you could end up with cell doublets. So we do doublet scoring as part of the single-cell analysis, and then you can adjust which threshold you want to use to detect doublets. 0.5 is the threshold that we have set as default, but obviously you can increase that or decrease that depending upon your specifications. And once again, you have these settings over here which control the dimensions of the visualizations that are generated. The step over here controls how many features you want to use for integration. Obviously, integration only comes into play when you have multiple samples that are part of the same experiment. Otherwise, this setting is not utilized within the pipeline. Now, these settings over here are quite important because these control the genes that you want to be visualized from your annotated RDS object or your annotated single cell object that you can then use for finalizing the annotations on your end. So this is a PBMC data set. I am interested in tumor-infiltrated leukocytes cell populations. So I'm looking for different cell populations within that subset. So some of the populations that I'm interested in are, for instance, naive, precursor exhausted, effector T cells, and others. So for these, what you can do is just add the number of genes or the name of the genes that you want to look at for this specific cell annotation. And then what the pipeline will do is generate dot plots for all of these genes. If some of these genes are not present within your single cell object, those will obviously be excluded from the visualization, but the genes that are present will be visualized in dot plots. And the way dot plots are generated is that depending upon each gene list that you input that are separated by these semicolons, the pipeline will generate individual dot plots containing these genes. So for instance, I have listed 4 different gene lists over here. So gene memory naive revisited, gene exhausted, precursor exhausted, and effector. So this pipeline will generate 4 dot plots separately that contain these genes. And obviously you can expand on this or reduce this as necessary. And there's a few different places where you can enter the genes of interest. So this is for preliminary marker labeling. So for instance, the cell populations in my samples, they're PBMC. So one of the first populations that I want to look at are CD4s and CD8s. So this will generate a really nice violin and dot plot containing these genes visualized for each cell cluster. And there are a lot more advanced settings over here that control, again, once again, the dimensions of the plots that are generated. These settings are also quite important. So for instance, this setting over here controls the mitochondrial DNA cutoff that you want to set. According to literature, the default that is recommended is 5% mitochondrial cutoff. So any of the cells that contain greater than 5% mitochondrial DNA will be excluded from further analyses. You can obviously increase that to 10%. More than 10% is not recommended because that probably will capture a lot of cells that are dying or not healthy. So 5% is the industry standard, and we've set that as the default over here. This setting over here controls the minimum number of features that must be present within each cell for it to be included in further analyses. Default of 100 is appropriate. According to industry standards, anywhere from 100 to 300 can be used. We've set 100 as the default, which allows us to capture a vast number of cells within our data set. This setting just controls the number of principal components that we want to use for nonlinear dimensionality reduction, which ends up generating the UMAP. So 30 is the default, and 30 is what's recommended by literature as well as industry standards. And again, you have some other settings to control the dimensions of your plots. This setting controls the normalization, the number of features you want to use for normalization using SC transform. And so forth. So once again, you have access to over 80 different settings within your advanced settings drop-down menu. And you can collapse it or expand on it as necessary. And one thing that we have done is that for each of the setting within the advanced settings, you have a detailed description of what this setting entails and what it controls. But again, if you have any questions, we also have a extensive documentation on the pipeline in the About section. Okay, so my advanced settings look appropriate. At this point, I'm going to click Next and select the appropriate speed tier. These are human samples, so I'm going to select the human speed tier and then click Next. I'm going to assign the run name. I'm going to say single cell RNA-seq test 2. And then I'm going to confirm that my samples that I selected are listed over here. And then, as you can see, the pipeline speed tier, I selected human, which is appropriately listed over here. This over here gives me the appropriate estimated runtime for run completion and the number of credits that will be used to run this pipeline. So at this point, I can click Launch Pipeline. As you can see, the pipeline is currently running. We will return once the pipeline finishes running. Okay, the pipeline has completed running. And at this point, I'm just going to do a quick walkthrough of the output directory. One of the first thing the pipeline does is generate a multi-QC report. So since we start from fast queues, the pipeline first aligns the files to the appropriate reference, and then reports some metrics in the multi-QC report. So you could just visualize the multi-QC by opening it directly within BPE. And this will allow you to understand the quality of your fast queues as well as some of the alignment metrics. All of the outputs are placed in this test folder. So I'm going to click over here. And as you can see, the pipeline generates a variety of different outputs from each step of the single-cell RNA-seq analysis, starting with alignment all the way to DG analyses. So if you remember, during the discussion on the advanced settings, we discussed how to set the appropriate resolution parameter. We set the default as 0.5. But if you go into this folder over here, Surat dim redux, this will contain a UMAP with all the different resolution parameters that we specified. And based upon this UMAP plot, you can determine whether the resolution of 0.5 was appropriate or not. And then you can attempt to rerun the pipeline with the appropriate resolution parameter. So as you can see, resolution of 0.5 generated 11 clusters, and the different cell populations are clustered apart from each other. So this resolution parameter was likely appropriate for my analysis, and then I'm going to continue to use this resolution for this specific data set. Another thing to note is the marker gene analysis. So as you remember, we discussed how to enter the appropriate gene list for marker gene analysis. If you remember, we specified different gene panels that we wanted to look at in order to finalize our annotations. So over here, different dot plots were generated for each one of those gene lists. And if you open these, as you can see, it generates really nice dot plots, which will allow you to determine the appropriate annotation for each one of your subclusters. As you can see, cluster number 6 is very high in GZMB, which is a strong marker for effector T cells. So using the different gene panels that we specified, we can finalize the annotation of cluster number 6, which likely corresponds to effector T cells. And obviously, you can run through the different dot plots that contain different gene lists that we specified to finalize the annotations of your cell clusters. You also get different dot plots from the automated annotations. So one of the automated annotations we use is called Azimuth. Going into that folder, as you can see, it generates an RDS object, which is a Seurat object that contains these annotations. The plots are contained in this folder over here. You can simply open this plot, which will allow you to visualize the UMAPs that are generated. The Azimuth annotation engine generates annotations of different granularity. So this is just the entirety of your Seurat clusters over here. This is the less granular or the top level annotations generated by Azimuth. And then this is the more granular annotations that are generated. As you can see, you have multiple subpopulations that are labeled over here. We also do annotation using Project TILS, which contains a very validated and strong panel for CD4 and CD8 TILs, or tumor-infiltrating leukocytes. So you can also visualize those annotations over here. And over here. The DGE analyses or the differential gene expression analyses is included over here. Over here, you have a CSV file which contains the differential gene expression list across different cell clusters. This is a large file, so you're not able to open this within BPE, but you can download this to visualize all the differentially expressed genes across the different cell populations in your data set. Last thing to note, we also generate a UMAP density plot. So if you have multiple samples within your data set and you want to get a visualization of population shifts across different treatments, this can be very nicely visualized using this density plot. And this just goes to show where the cell populations are concentrated within your data set. So this red indicates that within your data set, most of the cell populations or the cells tend to be concentrated within this specific cluster. And once again, this allows you to visualize the cell population drifts within your data set. So this has been a demo of the single cell RNA-seq analysis pipeline. I hope you enjoyed this demo. If you have any questions, please feel free to reach out. Thank you.